Hello, welcome to 13 Universal. This is A-level CIE Biology with Shedro. So we are looking at all topics theory paper 2, series 5. Okay, we have done series 1, 2, 3, 4. This is 5. Guys, I want to tell you that all these questions come from the past questions of mass scheme or CIE, right? So that's why we took these questions to solve. So don't forget that you must have read the topic, understood the topic, all right? And then you're ready to apply concept. Let's go. Let's apply some concept. Bring your writing material, your writing pad and pen. Let's apply some concept. All right. It says blood contains white blood cells and red blood cells. Yeah. Fig 3.1 is a photomicrograph. That's micrograph picture from light microscope showing different types of red blood cell. You can see different types of red blood cell. Okay. Of course, these are what are these? These are red blood cells. Okay. So these are white blood cells. Okay. All right. Let's see what the question wants us to do. Identify cell X and cell Y in fig 3.1. Okay, so identify what is cell X and Y. Tell me why, because another question can ask you what is cell Y. So pause the video and write it down. Write cell X is dash, cell Y is dash. Write it down quickly and let me know when you're done. Okay. Thank you. All right, guys. Cell X is actually a neutrophil. How do I know it's a neutrophil? Can you see the nucleus? The nucleus is irregular. Neutrophils have irregular nucleus or lobed, L-O-B-E-D, lobed nucleus, irregular. Now that helps them to squeeze through the endothelial pores of capillary wall into tissues when there is any invasion by pathogen, right? So of course, if you check, you see, all right, granules or this dot granules, right? Okay. Now, but is the nucleus the focus on? All right, it's lobed. So X is neutrophil. This looks like neutrophil. Now, if you look at Y looks like a monocyte. Monocyte, the mature one is called macrophage. Why? Number one is that the nucleus is kidney shaped or bean shaped. Okay. Now, it could be confusing. This macrophage or monocyte should be the first thing to look at because it is the nucleus is kidney shaped, all right, or bean shaped. But if somebody mentions lymphocyte, it appears like lymphocyte, but not exactly. Why? Because in lymphocyte, the nucleus occupies about over 2/3 of the cytoplasm, right? This seems to look like that, okay, but it's more likely to be accurate to be called macrophage because of shape of the nucleus, right? So let's look at what the mass scheme says. So mass scheme says X neutrophil, that's correct, monocytes. Okay, if somebody mentions lymphocytes, said accept it. Why? Because the nucleus appears a bit compact, and occupies a lot of space. But don't forget that for monocyte, it's kidney shaped. Okay. All right, let's go to the next question on this. So it says the width of one of the cells in fig 3.1 is represented by the line Z. Okay, the actual width of line Z is 8 micrometers. Okay, calculate the magnification used to produce the image shown in fig 3.1. Write down the formula you will use to make calculation. All right, so this is magnification. Don't forget, guys, of course, um, remember the formula of magnification. So magnification, write it out, is equal to what? What is magnification there? Remember the formula, image size over actual, right? Yeah, so the image size of actual, right? But you see, if you want to calculate this, you make, because what we're looking for here is magnification. Yes, that's magnification. So what do? Go back to the diagram and measure that line Z. So that is image size. So that line Z in actual paper, when you measure it, the line Z is our image size, and it is about 1.2 centimeters. Okay. Yes, so measure it is about 1.2 centimeters. So measure it with your ruler. So when I did it in the actual paper, you may not get exactly this, but 1.2, right? But you see, we work with micrometers. Okay, micrometer is the basic unit we use to work, especially when dealing with a cell. So how do you convert 1.2 centimeters to micrometers? So what is the conversion factor, guys? The conversion factor, of course, is 10,000, right? So you multiply the value with 10,000 to convert it to micrometers. So that will be 12,000. Is that okay? So 12,000 is our image size. So what should be our magnification then? So that is image size over actual. Actual is 8. Okay, so get me your calculator, punch your calculator. 12,000, okay, divided by 8. What do you have, guys? Of course, 1,500. So this is going to be times 1,500. Okay, so always measure. When you measure the image, convert to micrometers, and that gives you the image size. Okay, so that is it there. That's 3i. So this is what the masking says. All right, magnification is image or line z or length or width all over actual, or this is acceptable. This is the one I used here. Or so this is it. Any of this range is correct. Okay, or use the triangular method. You know the triangular method. Okay, let's go to the next question, guys. Carbonic anhydrase is an enzyme found in red blood cells. You know carbonic anhydrase? Say describe. Anytime they say describe, tell us what and how. How. Okay? Describe is a command word. Tell us what and how carbonic anhydrase is involved in the release of oxygen. How is it involved in the release of oxygen from red blood cells at respiring tissue? How is it involved? Okay? Now before we bring up the answer, I want you to pause the video and try solving it by yourself. Okay? Try solving it by yourself. All right. And then you continue. So remember that we are practicing here. So we'll try to practice. So please make sure you are applying the concept, because you only learn applied skills by solving questions. Okay. So let's assume that this is the wall of capillary. Is that okay? The endothelial cells lining the capillary. I just want to use it to show you something. I've done this before in other questions, but just to show you something. All right. Now this is also this is the wall, the endothelial cells. In between them have pores, endothelial pores, through which blood plasma leaks out to form tissue fluid. Okay, now this is, I want to draw the red blood cell. Okay, the red blood cell most times, right, in the capillary. So these are tissues here, right, tissues here. So remember that don't forget that capillary is a site for exchange. Okay, that is where exchange occurs. Let me paint the picture here and then we'll solve the question. All right, this is the image that should be in your mind, because we use imagination a lot in biology, okay, to solve questions. Imagine this, right? Use imagination in your mind as you're thinking. This is what you should be thinking. Okay, you can write it out if you like, but it's going to take you time, but imagination can help you. All right, so remember that this is respiring tissue. So CO2 diffuse from these cells, pass through the endothelium and then enter the red blood cell. Can you see? So CO2 basically we react with water, remember? So this is red blood cell. Inside red blood cells, CO2 combines with water. You remember this equation? Okay, so when it combines with water, what does it form? So it forms, remember this reaction is reversible. So the fourth reaction is in respiring tissue. So what it forms is this acid called carbonic acid. Is a weak acid, right? So H2CO2, that's carbonic acid. Carbonic acid is formed. All right. So, and then of course this is going to dissociate to give us hydrogen ion. Okay. So hydrogen ion is formed when it associates and another ion, okay, that is hydrogen carbonate ion. Don't forget that. Okay, so hydrogen carbonate ion is formed, but you see hydrogen carbonate ion is transported in the plasma, so it's going to move out of plasma. All right, now that's a negative ion, right? So remember, the forward reaction occurs in the span tissue. Backward reactions will also occur in alveoli when the blood gets to the alveoli. Okay, so you know the story. You know the story how this hydrogen carbonate ion diffuse into the plasma to be transported, and then to balance the electron neutrality, because when negative ions moves, it creates an imbalance. Chloride ion moves in. Chloride ion moves in from the plasma into the red blood cell. Okay, that's chloride ion. All right, chloride ion moves in, and that's called chloride shift, guys. So now let's face our question. Now let's get back. Say, describe how carbonic anhydrase is involved in the release of oxygen from red blood cell. Okay, don't forget that, guys, quickly, that while here, that oxyhemoglobin, of course, does something. It has to unload oxygen, right? So oxyhemoglobin, remember oxyhemoglobin, write it out. Okay, that is this. Yes, so it has to dissociate. Okay, yeah, we solved this question before. Something like that, not exactly, to give us hemoglobin and oxygen. So hemoglobin is Hb, right, plus 4 molecules of oxygen, right? Yeah, okay, you remember the formula? Okay, so you see it unloads it, right? So this hemoglobin will now be able to combine with something, right? It combines with that hydrogen ion. Remember that hydrogen ion could change the pH of red blood cell, because hydrogen ion lowers pH, it forms acidic pH, and the pH of red blood cell is actually neutral. So hemoglobin acts as a buffer here. All right, hemoglobin acts as a buffer. He's a good guy. So it combines with hydrogen ion. He said, look, let me save the day, because if the pH changes, there's going to be a problem. Change in pH denatures carbonic anhydrase enzyme, okay, and then the red blood cell is in trouble, and then we are in trouble. So hemoglobin is our hero. Come on, somebody clap for hemoglobin there. All right, so let's look at how this plays out. Come, let's go to work. It says, describe how carbonic anhydrase is involved in the release of oxygen from red blood cells. So you see, carbonic anhydrase catalyzes this reaction. So what you say is that, because they say describe, so step by step, how does it release oxygen? How does it release oxygen? So what happens with carbonic anhydrase, how it starts is that it catalyzes a reaction between CO2 and water. It catalyzes this reaction to form carbonic acid. Okay, so carbonic acid then dissociates to form hydrogen ion and carbonate ion. All right, so hemoglobin then releases oxygen. You see, oxyhemoglobin unloads oxygen. Why? In order to bind with hemoglobin. Okay, but the question says how. Okay, now hemoglobin and oxygen, they bind to, all right, to the hem group, all right, or close to each other, but the same site. So that means hemoglobin cannot bind to oxygen and to hydrogen ion at the same time. It has to bind to one. So that means, how does it do? So oxyhemoglobin has to release oxygen in order for hydrogen ion to bind with hemoglobin to form hemoglobinic acid. Okay, so what is the role of this here? I've said it. It catalyzes this reaction. This is where carbonic anhydrase plays a role, both forward and backward. Okay, helps this to bind to this in order to form carbonic acid, all right, which then forms hydrogen ion and hydrogen carbonate, okay, ion. And then once this is formed, hydrogen ion then of course need to bind to hemoglobin. So hemoglobin will release, oxyhemoglobin releases oxygen, unloads oxygen, so that hydrogen ion binds to the same site, all right, or causes hemoglobin to release oxygen, so they can bind to the same site. So we've answered this question. It's about 4 marks question. Okay, so what do you think? Let's look at what the mask says. Any 4 from this. Okay, it says, look at this: carbonic anhydrase catalyzes the reaction between CO2 and oxygen. That's what I said here. Okay, any other wording? AW means any wording. Okay, now the next one is to form carbonic acid. Yes, you see, sequence by sequence, because the command word says describe. When you hear describe, you must consider sequence, guys. Okay, to form carbonic anhydrase, what's the next that happens? Okay, hydrogen ion forms. Look at it. This dissociates to form hydrogen ion. Okay, it forms with this from dissociation of carbonic anhydrase. So hydrogen ion then binds to hemoglobin to form hemoglobinic acid. That's it here. It binds to. Now how does it do that? It displaces, okay, or releasing oxygen to form hemoglobin. So it releases oxygen from hemoglobin to form hemoglobin. All right, so you see, so now they bring the concept of allosteric site, change the tensile structure. Oxyhemoglobin maybe changes its shape, causes release of oxygen, and then binds to hydrogen ion. That's it, guys. So look at it, memorize this mass scheme and understand it and memorize it. Biology deals a lot with memory, and then connect it to the sequence. Okay, you answer question based on the command word, right? Command word says we should describe as a sequence to that. Okay, let's move to the next question. Sulfonamide is a competitive inhibitor. Carbonic anhydrase is an enzyme found in red blood cells. Fig 3.2 shows the effect of increasing substance concentration on the rate of reaction catalyzed by carbonic anhydrase. Sketch a curve on Fig 3.2 to show the effect of sulfonamide on the rate of reaction catalyzed by carbonic anhydrase. Guys, yes, I want you to sketch it on all right, the graph. What do you think? All right, is the answer. Sketch it. Try it. Remember that sulfonamide is a competitive inhibitor. So when you continue to increase substrate concentration, what happens to reaction that has competitive inhibitor? This one now has no, all right, has no inhibitor. This is the first graph. But by the time we add competitive inhibitor, increasing substrate concentration will reverse it. So the graph will be under the main graph. Okay, it will increase, the reaction will increase, and then reaches to where it plateaus together. All right, sorry for the line is not straight. Okay, let me try to see if I can do something a bit straighter, but you understand what I'm trying to say. Okay, it goes and then reaches where it is. Okay, and that is why you see that with competitive inhibitor we're going to have, all right, the same Vmax. So increasing substrate concentration reverses the effect of competitive inhibitor. So we have Vmax. See, Vmax will be the same. All right. Yes, Vmax is the same with competitive inhibitor. All right. And of course, if I decide to find the Km, which is not part of this question, but we're doing revision. So Km is substrate concentration at half Vmax. So all I just need to do is divide the Vmax, all right, by 2, then come to half Vmax. All right, and then find the substrate concentration. So this is still Vmax. How do I find the substrate concentration? Trace it to this graph. Right. Okay, so now trace it to the main graph without yes, and then take it down, extrapolate it. Right. Okay, take it down. So this is where Km is. Okay, so you see that but if you see with competitive inhibitor, if I extend it, all right, the Vmax of competitive inhibitor will always be higher than the Vmax without competitive inhibitor. But guess what? They will have the same. Sorry, the Km. I'm talking about Km. The Km of competitive inhibitor. That's what I'm doing now. Will always be higher than the Km of without competitive inhibitor. Now this is Km with competitive inhibitor. All right, it's higher, but their Vmax is the same. But remember, if you are dealing with non-competitive inhibitor, the graph will go like this. Graph will go like this. It will go like this. Okay, it will not reach the Vmax. So this graph is for non-competitive. This is competitive. This is without competitive or non-competitive, without inhibitor. All right. So with competitive, same Vmax, but all right, higher Km. All right. But with non-competitive, the Vmax decreases, but it's going to have the same Km. All right. So the answer says, because they say sketch a curve. They said the curve below the original line. So for computing inhibitor, this is this one, okay, will be below the line. All right, we merge with the original line after it begins to plateau. So you must merge it with it here. All right, that's all you're needed to do. Food crops such as barley and wheat contains gluten. Gluten contains two proteins, gluten and gliadin. Let's tackle this question. Table 4.1 contains description of the structure of gluten and gliadin. Okay, gluten and gliadin, they are, all right. You know, gluten is what makes bread stretchy, gives bread characteristic look. I don't know why you like bread, but bread is sweet, man. It's good. So gluten, that's what we're talking about. Table 4.1 contains description of structure of gluten and gliadin. Complete table 4.1 by writing the levels of protein structure that applies to each of these description. Okay, look at it, guys. Try this question. See description. What level of protein structure? Gliadin protein is a single polypeptide that forms compact structure. What does it tell you? Compact structure is same as 3D, and that's tertiary, right? 20% of amino acid gliadin molecules are glycine. So which level of protein structure talks about amino acid, okay, sequence or order of amino acid? That's what? Primary. Gliadin and gluten molecules contain regions of beta pleated sheet. Which level of protein structure talks about beta pleated sheet or alpha helix? It's called secondary structure. Yeah. All right, let's hear your questions. Want to hear your questions? Share this video, make comments, okay? All right, let's see. Tell us what you want. Somebody mentioned they want more topical questions from immunity, you know, and the rest of them. We got you covered. We're going to do that, right? And we're going to bring other subjects too in the future. Many genes in eukaryotic cells contain introns. The genes that code for gliadin do not contain introns. All right, do you understand what introns are? Remember, in biology, the most important skill in biology is memory. All right, reflective thinking. If you can't remember intron, you can't do this question, guys. That is why when you're studying biology, remember, study to understand, but also study to remember. Okay. All right, now introns are non-coding sequence. They are non-coding sequence. So that's what we call introns. Some of them are regulatory sequence that help to regulate the expression of genes. All right, look at it. Let's explain. Tell us how, with reason, a lack of introns in gliadin gene affects the production of messenger RNA from the primary transcript. How will it affect the production? Okay, it's going to be exactly like the other ones. Okay, remember, I don't want to spend more time on this question. All right, so this doesn't have it doesn't have it doesn't have introns. Okay, so you see, during transcription, remember that a segment of DNA will unwind where the gene is found. Okay, a segment of DNA will unwind, and of course I'm not going to talk about transcription. I'm going to show you something. All right, so the template strand is used to make the primary transcript. The primary transcript is made, right, using the template strand. Okay, but this primary transcript will, normally what happens is that after transcription, all right, it's going to be converted to messenger RNA. This is the messenger RNA, the functional mRNA here. All right, the first one there is primary transcript. All right, so what happens is what we call post-transcriptional event happens, where introns are removed, exons are joined. Exons are coding sequence. All right, but here the question is explain how a lack of intron affects the production. Simple. There are no introns. Okay, we don't have there are no non-coding sequence, so there is no splicing, so introns are not removed. Okay, exons are not joined. So if introns are not removed, exons are not joined, that means that our primary transcript codes for the same sequence as the messenger RNA. So the messenger RNA, the primary transcript here, this first one, and the messenger RNA will be the same. There is no post-transcriptional event. That's what is going to happen. Now, well, let me say it again. All right, that there will not be any introns, so no post-transcriptional event, no splicing, so introns will not be removed, exons are not joined, so the primary transcript will code for the same sequence as the messenger RNA, so they'll be the same. All right, and of course, let's see what the mass scheme says. All right, look at what masking says. It's RNA splicing not needed, does not occur. Yeah, reference to all primary transcript is a coding sequence. All primary transcripts here, coding sequence. Okay, no nucleotide removed from the primary transcript, all right, because there are no introns after transcription. No process of exons joining, no non-coding sequence, so introns, all right, not need to be removed. Or any alternative wording, only one type of mRNA form, okay, either that post-transcription event does not occur, all right? So there's no need for all of that. All right, let's go to the next question. Celiac disease is a condition in which the immune system of a person responds to gluten in their diet. So people react to this gluten in their diet, all right, maybe vomit, their stomach get bloated up, all right, immune response, temperature rises because they just ate something. So the celiac disease is a celiac disease condition in which immune system of a person respond to gluten in their diet. Let's see what happens. In celiac disease, there is a response to the presence of peptides. Peptides, short chains of amino acids that are produced as a result of gliadin digestion. So the first question says, remember, understand the background information. The context is very important. You apply your content to match your context. Your context is the background question. There's what is called context content matching, but you have to understand the content. All right, the gliadin peptide produced as a result of digestion are often as large as 33 amino acids. That's too large. Okay, intestinal cells take up large number of these at the same time. All right, so that's large distance. Okay, so the question here is, how does, I think something is missing. It's asking you, how does the cell take up something as large as 33 amino acid? How does it happen? How does cell, by what process does the cell take up bulk molecules, right, from outside the cell to the inside the cell? Movement of bulk molecule from outside to inside is endocytosis. Okay, so endocytosis, let's assume that this is the cell, you know it. Okay, so this is the bulk molecule, right? So what happens, guys, during endocytosis is that there's going to be an engulfing. So the cell membrane of this cell invaginates or folds inward round the molecule that's been transported. It invaginates, okay, folds around it, okay? And once it folds around it, remember that cell membrane is flexible, so cell membrane will rejoin, all right, okay, to form what? To form endocytic vesicle. So you see, right, cell membrane folds around the molecule we are moving inside. Okay, so that's endocytic vesicle. So this is of course, let me say it, all right, so engulfing. How does it move it inside? This is going to invaginate, cell membrane invaginates to move this molecule gluten, right? How does it invaginate? Move, folds around it, and then because it's flexible, it can just rejoin here. It rejoins to this to form a phagocytic or endocytic vesicle or pinocytic vesicle. All right, remember that the formation of this vesicle requires ATP, so it's an active process. Okay, so let's see. All right, so endocytosis, yeah, pinocytic. So what's the difference between this and this? This is movement of solid molecule. This is liquid. All right, cell eating, cell drinking, pinocytosis. Detailed invagination of a membrane, uses of ATP, active process energy is needed, formation of endocytic vesicle. All right, energy is needed to form the vesicle. Okay, let's look at the next question. The presence of gliadin causes the immune system of a person with a celiac disease by producing antibodies. So immune system will respond by producing antibodies. All right, so how does that happen? Describe the sequence of events that result in immune response producing anti-gliadin antibodies. So how does that happen? Pause this video and try to solve this question by yourself. Try to solve it by yourself. Okay, first of all, apply it. How does, now, what are the sequence of events that occur during immune response? Okay, that's what they're asking you, to produce antibodies. Okay, the first thing that happens, guys, is called antigen presentation. Macrophage can do that, or infected body cells. All right, antigen presentation. So macrophages present antigen on its cell membrane, okay, to lymphocyte. Then the second thing that happens is called clonal selection. Okay, clonal selection is that the lymphocyte with complementary receptor to the antigen. In this case, the antigen here is gliadin. Gliadin is the antigen, is non-self, foreign. So when it's being presented, not all lymphocytes can recognize it. The only lymphocyte that can recognize, so recognize means to bind, all right? The only lymphocyte that can recognize it is the lymphocyte that has complementary shape to it. I'm tempted to draw this. I don't want to draw this because of time, okay? But you know it, all right? So if this is presented, it's only lymphocyte that has complementary receptor that can recognize it. So this is gliadin, right? Maybe being presented by a macrophage here, right? A macrophage is presenting it, right? So there are other lymphocytes, okay? Other lymphocytes that are here. For example, this is another lymphocyte, but their receptors are different. They're different clones, okay? Yeah. So let me give you another type of lymphocyte here. But what differentiates them is their clone, is their receptor. Yeah. So do you get? So it's only one. So antigen presentation, this presents this antigen, right? And then this is the next is clonal expansion. So antigen presentation, antigen selection, then clonal what? Expansion. So this cell, this particular clone, divide by mitosis to form other cells. So clonal expansion occurs. They divide rapidly to produce more cells that are like it. And then that's clonal expansion. And then if the cell is a T helper cell, the T helper cell will respond by secreting cytokines. Cytokines will now stimulate appropriate B lymphocytes to now divide and then secrete antibodies. B lymphocyte divide, appropriate lymphocyte divide, all right, and then differentiate into memory cells and plasma cells. The plasma cells now secrete the antibodies, all right. But if it is B lymphocyte that this is meeting straight, it will divide and then differentiate and then secrete antibodies. Don't forget, the first is antigen presentation by a macrophage, all right. Then the second is clonal selection. Now these are lymphocytes. Only the lymphocyte with receptors that has complementary shape to this antigen, this is the gliadin, right, will fit into it. This can fit it, this can fit, right? And then the one that fits in leads to clonal expansion, division by mitosis. And then if it is B lymphocyte, it will differentiate to form plasma cells, and memory plasma cells secrete antibodies. That's how antibodies are made, okay, against, all right, gliadin. All right, let's see the mass scheme, guys. See what mass scheme is. NE4. Reference to non-self, okay? So gliadin is non-self or foreign. Gliadin acts as antigen. So reference to antigen presenting by macrophages. So this is acting like macrophages, just recognition activation. So this is recognizing antigen clonal selection. Clonal expansion by lymphocyte or T lymphocyte, so they divide by mitosis. You can see. So T helper cells will secrete cytokines. Cytokines will now stimulate B lymphocyte, which is called humoral, okay? All right, that will form plasma cells that secrete antibodies. So guys, this is where we stop for today. All right, so let us hear your comments. Please ask questions, make contributions, share this video, like us. See you next series 6. Bye. This is Shedra.